We've all been very focused on how to stay healthy these days, but we've not been talking about hydration. If you get coronavirus, flu, or even experience allergies, cold, a variety of everyday ailments, they all need hydration. And that's why it's a perfect time to welcome our friends at Hydrolyte back. This is a great product. You all know I've talked about it for a long time. This was the hydration product I wanted to invent, and they got it there before me. Now, remember, dehydration can make you feel sick, even a slight amount, and none of us need that anxiety right now. So stay well hydrated. I am thrilled to welcome our good friends at Hydrolyte back to the show. Longtime fans remember my obsession with Hydrolyte, which is literally the best hydration product I have found. I'm even more excited to introduce their brand new single-serve powder sticks. Simply pour one powder stick into a glass of water. They recommend 7 ounces. The powder dissolves instantly and creates the perfect balance of sodium, glucose, and water to deliver up to 4 times the electrolytes of your typical sports drink. And think about it. You can take this anywhere. You should have it on hand to just pour it into water, and you have a real significant hydration product. The other great news about Hydrolyte, the powder sticks, they are 100% natural, no artificial flavors, colors, or sweeteners, and they are available in flavors like orange and lemonade, and they taste great. Hydration is crucial. Hydrolyte is the fastest and easiest way to stay ahead of it. And you can find Hydrolyte powder sticks in the digestive aisle at Walgreens or Amazon, or simply just go to my website, drdrew.com/Hydrolyte. Again, that is drdrew.com/HYDRALYTE. You see it spelled here. There it is. And you enter the code DrDrew18 to save 30% off your order. Forget the runs on toilet paper. There should be runs on this. This is doing much more for your health than toilet paper. So go get some. Have it on hand. If anybody in your family gets sick, you need to keep them hydrated, and that's how you do it.
Hey everyone, it's a dose of Dr. Drew.
It is a dose, a very special dose. It's a very special dose indeed. We're going to talk biology today with Dr. Yogendra and Dr. Patterson. I will give you their particulars in just a minute, but I just want to remind everyone that today is brought to you by Hydrolyte. Go to hydrolyte.com/Dr. Drew and use that code Dr. Drew 25. There it is. Wish I was going to invent this.
It's actually drdrew.com/Hydrolyte.
Oh.
My bad again.
Okay, well, it's backwards. But be that as it may, that's a product I wanted to invent. They got there first, so I'm standing behind them. So here we go. Let me tell you about our guest. This is a very special day. I see you guys lining up on the chat. I want to give you a chance to pile on in here. Somebody tweeted something very appropriate, which was bring the real deal today. And I thought, yeah, today is the real deal. We are being revisited by Dr. Ram Yogendra. He's a master in public health and an M.D. and an anesthesiologist. Background in tropical communicable diseases from University of South Florida, M.D., and now practicing anesthesiology and also the medical director of ECA Wellness, a sports and medical fitness center. And we are very privileged to welcome Dr. Bruce Patterson in here. Let me give you more of Dr. Patterson's particular undergraduate in molecular biology. Guys, this is sort of funny to me, thinking 3 biologists walk into a room. You know what I mean? Because I feel like trained biologists are not out in the media very much today, and that's part of the problem with all the craziness and all the politicization of everything that's going around about COVID, but I digress. He received his molecular biology undergraduate degree from Michigan, Northwestern M.D., very instrumental in the HIV and AIDS epidemic, many publications, including over 150 manuscripts and books focusing on single-cell biology and diagnostics, formerly associate professor of pathology and infectious diseases and director of virology at Stanford, currently serves as CEO and founder of IncellDx, INCELLDX, Inc., a growth stage company that has translated research into discoveries, state-of-the-art single-cell diagnostic, cancer, immuno-oncology, and infectious diseases. Gentlemen, thank you for being here.
Thank you. It's a pleasure.
It's really our pleasure.
Thank you, Dr. Drew.
Yeah, and Dr. Yo, thank you for bringing Dr. Patterson. You threatened you would, and you delivered. So thank you for that. Before, I didn't realize both of you had such extensive background in infectious diseases, particularly in tropical infectious diseases. We are on the eve, before we get into the whole story we're about to get into in terms of COVID and cytokine storm, I just want to dispense with this that is on my mind. So much is being not addressed because of the singular focus on COVID. And we are on the eve of a plague outbreak in Southern California. We have had the most massive rodent blossom of all time, literally millions and millions and millions of rats. And now we have our first human subject of bubonic plague up in Tahoe. We've had 3 years of typhus here. And I've been saying that if we don't do something about the rats, the only thing I know of that follows typhus is Yersinia. Either of you have an opinion about that? Am I right?
Yeah, absolutely. And I think we have to prepare ourselves a lot better as a country for emerging infections or reemergence some of the pathogens of old that you hear in, great novels by Dickens and such. But we really have to have a strategy that is focused on emerging pathogens that may not be pathogen specific. And I think that's why we're here to talk today, is that we need strategies that when we're faced with pathogens that our body has never seen before, our bodies don't have that memory that we usually do against other pathogens in the world that we commonly come in contact with. And we have to have a broad spectrum infection strategy that is not pathogen specific.
And I think people may becoming certainly they're becoming familiar with respiratory spread. That's obviously why we're all wearing masks. Before it became clear that COVID was almost exclusively spread that way, we were thinking about it in terms of other viruses being transmitted on our hands. But what worries me about the plague is we're not used to talking about vector transmitted with things like rodents and our dogs and our cats, and that's all of these have to be planned for, correct?
Right, and all the people who have suffered from mosquito-borne
disease, yes,
and ticks who are still suffering, and it's not unlike what we're going through right now.
All right, so Dr. Yogendra, you're sitting quietly right now. Let me give you a chance to outline your theme for today that we're going to get into. Is it all roads lead to RANTES?
Yeah, it's all roads lead to RANTES. And again, thank you very much for having me on again. The last time we sort of talked a little bit about RANTES as being that chemokine, specialized cytokine that Dr. Patterson's theory is playing. It's the major this is the quarterback of the cytokine storm in that immunological phase. What's interesting is when Dr. Patterson started talking about RANTES a couple months ago, I went to those immunology textbooks and virology journals, and everything he was saying was illustrated clearly in those textbooks. So it wasn't anything he just pulled out like some magic sheet of paper he pulled out of the printer and something he just thought of. This is in the literature. And what's really interesting is we're starting to find out that RANTES, and this is what I think Dr. Patterson's really going to get into, is how everything connects, not just COVID, but cancer, HIV. And we're also seeing there are many drugs now that are being developed. They're showing a lot of promise. Last time I was on, we talked a little bit about leronlimab.
Yeah.
But there's another medication called Abitadel, that's RLF 100. And we just found there's some literature out there from Spain suggesting that the drug actually possible main mechanism of action is on RANTES. I sent the journal article to the researchers with Abitadel, and now they're going to start looking at RANTES. Dr.
They thought it had something to do with the surfactants in the lung, but I was looking at that too, and there's a lot of buzz about Abitadel as well out there, and I think you're right. I think the issue, and not only is it going to change cancer and other, of course, rheumatic diseases and things like that, but I just keep thinking that the phenomenon of intensive care medicine is going to be changed forever, immediately, and these sorts of interventions are going to have lots of application. So. RANTES and by the way, you're right about the CCR5 and CCR5 stuff. It's all out there in the literature, but it wasn't in the common lexicon. The average doctor walking through an ICU was not thinking about these things, right, Dr. Yo?
Yes, absolutely.
Yeah.
And and when Dr. Patterson and I started talking a couple of months ago, I told Dr. Patterson, when I was in medical school, I heard a little bit about this CCR5 and being in HIV/AIDS, sort of on the public health side of things. You hear about this research. It just wasn't really talked a lot about.
Yeah, it wasn't in the clinical vapors. It wasn't just sort of, you wouldn't walk down the hospital hall talking about cytokine storm or activation even, right?
Exactly. And there were quite a lot of intensivists, and very early on in the pandemic were telling me that these cytokines are not elevated. They're not playing a role. Right. They're not having anything to do with long haulers. And here Dr. Patterson screaming at the top of his lungs that this is a RANTES disease and these are cytokines. And I feel like the media is probably about 3 to 4 months behind in what Dr. Patterson was talking about in March and April is what now, the New York Times and CNN and all these major media are talking about. So what Dr. Patterson's going to talk about today is probably something in December that the media is going to pick up on and talk about it. So I think it's just really important. I think it's really, I'm very grateful for you, Dr. Drew, because, we can sit and talk about amongst this, in our little medical circles of people that follow these drugs, but the American people need to know about what's going on.
Oh,
And
yeah.
there's a lot of fear mongering going on. People are terrified.
Oh, yeah.
And we'll touch upon it a little bit, in our discussion about the psychological effects that are happening. But again, it's just, I'm very grateful. I know Dr. Patterson is, that you have us on to it so that we can share this and get this message.
This has been my thing from the beginning, which is that I know us and our peers, and Dr. Patterson is the living embodiment of this. I know that we improvise and move and deploy, and we'll figure stuff out very, very quickly. So when what was the University in London, Oxford group was predicting millions and millions of deaths, I was like, not here. Not here. We talk about the weaknesses of the healthcare system in the U.S., not when it comes to mobilizing and improvising. We will figure this stuff out and make a massive difference. And Dr. Patterson, I thank you for being literally the embodiment of that. So let's go to that. Let's talk about. I hate to have you repeat your TED Talk. People can go to your TED Talk and hear the basics if they wish. How would you like to present this today?
Well, it's this is probably my fourth pandemic, and I started working on RANTES and CCR5 in the mid 1990s with HIV. When we found out that CCR5 was a co-receptor for HIV. Mm-hmm. And then over the years, as I got into cancer and through HPV infection and HIV and other routes, it became clear that CCR5 and RANTES had a role in cancer. And the fact is, it really is, a quarterback is a good name for it. Is it the only thing at play here? No, like a football team, you need the players around it. But what it does is it coordinates where immune cells go, how they traffic, and what their role is. So CCR5 can be on a certain subset of T cells that actually fights off virally infected cells or fights off cancer cells. Conversely, it can be on a cell type called T regulatory cells that inhibit that. So it's really, manipulation of CCR5 that could lead to the outcome that you want. But at the end of the day, RANTES, when you look at it, or CCL5, is responsible for the movement of immune cells. It's associated with metastases of tumor cells. It's associated with even renal failure with high RANTES causes inflammation, liver enzyme elevation. RANTES is made by activated platelets, which could explain the coagulopathies that we see in COVID. And frankly, the long haulers are a very interesting group, where they have this prolonged increase in inflammatory markers, IL-6, RANTES, IL-8. It's not where it was during the cytokine storm, but it's still there. And it's why they're feeling bad 30, 60 days out. And even compounds like TNF-alpha, which, back to your specialty, has been shown to cause depression and even mental illness in postpartum depression. So, I think what we're doing is seeing a lot of support for the naturopaths out there who have been espousing the role of inflammation in overall health for many, many years. And COVID is just bringing it to the forefront.
I'll grant you that. I was skeptical of the sort of hand waving. Inflammation is what's important, because it never zeroed in on the biochemistry until now.
Yeah.
And here it is. But you just packed a ton into what you just told us. So I want to go back around, and I want to
Sure.
sort of parse all this out a little bit. And I'm watching the thread here. Those of you on the in the chat stream here. So I'll look for questions and things there. I'll bring up to my guest as well. Before I unpack a little bit of what you said, I read your research, and I was surprised by two things. I expected all the effect on the cytokine system, and it all bore out exactly. I'm talking about leronlimab now. I'm talking about the publication you have on leronlimab, but we are talking about RANTES. And Dr. Yogendra, you had an interesting way of describing RANTES last time. like it was like a smell that sprayed into the room or something, and was it that how you? In my head, it stayed with me as that's the model of sort of molecules that create a smell that you follow.
Yeah, it's a chemotaxis, and whenever I read about chemo, I always make a story up in my head, all about analogies, and yeah, it's like perfume on one side of the room, and you're attracted to it.
Right. That's
so
what
if you put a little clamp on your nose, you're not going to be able to smell and go to that area where where the perfume is highly concentrated.
It that stayed with me. I thought that was a great analogy. But
it's immune cell pheromone.
Yeah, good. I like that. But back to the research. So I was surprised to see the degree to which the leronlimab had effect on macrophage and lymphocyte normalization. That jumped out at me as like, that is surprising. Did that surprise you?
Well, no, and you think about it a little bit like a war and sending troops in. I mean, basically what's happening here is you're getting a sequestration of all these immune cells to the lung because of RANTES, and it's really taking immune cells out of the circulation. But the other thing that was surprising to us was leronlimab was restoring or reversing immune cell exhaustion. So like any, either, sports or war, the troops get exhausted after a while. And so you have these immune cells that are fighting off this virus. And after, a week or two of exposure to high levels of virus, the immune cells become exhausted, so they can't function. We saw drops in CD8 cells which are the sort of the cells that fight off virally infected cells. That was analogous to the huge drop in CD4 cells, which is their compatriots in HIV/AIDS. So, these patients were hugely immunosuppressed and high at levels of cytokines. So if we could mitigate this mass migration through blocking CCR5, and if we could reprogram macrophages from producing interleukin-6 and other toxic cytokines that we've heard a lot about as part of the cytokine storm, the goal was that we could restore the immune response. And in fact, in the paper, the latest revision, there was an inverse relationship to the CD8 T cells, which are the ones they're the fighters. They're the ones getting rid of the virally infected cells. There's an inverse relationship between those going back up and plasma viral load going down. And I think that is really the take-home message, that our immune systems, as we learned from cancer, are perfectly capable of taking care of cancer virally infected cells when given the opportunity and when they're restored.
Do we think that this is a evolutionary strategy by the virus, or is it just something that just is an epiphenomenon?
Well, I think that's a great point. I think the viruses are amazing at their ability to sequester the host cells that they need to replicate. And we see that in any number of viral infections where, they sequester cells. A virally infected cell is not a normally functioning cell. So here was HIV/AIDS taking out, the key cell of the immune system. COVID doesn't take out the immune cells, but it infects the respiratory epithelium, producing cytokines and other mediators that do the same type of thing. The sum total is inhibition of the immune response. So then they can't fight off the virus. So they are elegant in their ability to, in an evolutionary aspect, sequester and inhibit those functioning parts of the body that will get rid of them.
Got it. You also mentioned the activated platelets, which I thought was a very interesting part of the story. That the coagulopathy associated with COVID is something that gets a lot of press and news. And from the medical standpoint, it's been a little mysterious. It doesn't respond to anticoagulants, doesn't respond to antiplatelets. But here now is a mechanism where you can go after it. Would that be accurate?
That'd be accurate. And what is really amazing is if you look at, the 700 HIV patients who have been, treated with leronlimab leronlimab, and also the patients in the trials have been treated with leronlimab, both in the clinical trials and in the EIND patients. Not a single one of them has had a coagulopathy.
That's
And I
interesting.
think
And
that,
if I remember the data correctly, in your control arm, so-called, I mean, I want to break that down a little bit. There was coagulation problems, right?
there was.
Yeah.
And in fact, one of the early criticisms of the paper was there wasn't a control arm. But again, we got into the game so early with patients who were absolutely critical. They were the worst of the worst. They wouldn't even be considered severe. They were worse than that. And again, they had everything under the sun. They had renal failure. They had, some of them had liver enzyme elevations. And then the control group had, coagulopathies, and they weren't treated with leronlimab. So, again, I think RANTES is something that, at this point you can at least hypothesize a role, but the fact is, if you look at the literature, it has a role in renal failure, and a role in liver enzyme elevation, and a role in coagulopathy through upregulation in activated platelets,
Right.
which bring in inflammation, and it causes basically a nidus for a thromboembolic type of event.
Right. I mean, we are people, maybe just to frame it, people understand platelet-rich plasma, PRP. They're using that now therapeutically. And so platelets have all sorts of factors that they produce. Maybe you can understand it that way. But the control arm of your study was the other thing that jumped out at me, in addition to the cellular restoration, which I found astonishing. But you went out of your way to create a control arm that was, I thought, very good. And the FDA is pushing back on that. I was shocked, because it was you documented everything you needed to document about that control group. It's just they weren't randomly selected; they were just selected, right?
Well, again, it was so early in the pandemic where we went to New York, and it was just a mess. There was lack of resources. It was mayhem. The ICUs were full. These patients were the most critical of critical, and we didn't even think about getting controls. I mean, the ethics of not giving a potential therapy to somebody who's absolutely critical never even crossed our minds. And then it really wasn't the FDA; it was actually the journals, when we submitted to them, reviewers asking, where are the controls? And the response is, you almost feel like responding, you know what, these people, there was no chance that you could keep therapy from a control population.
Yeah, the ethics. You got to go to an ethics committee and go, hey, where's your ethics board for this journal?
Yeah, exactly. But the fact is, we went to other colleagues who didn't have access to leronlimab. We found control patients. We did the exact same panels of sophisticated assays on that group, and finally came up with a control group that was acceptable, and things are moving forward now. And we're very excited about that. But yes, that was a huge challenge, finding a control group for patients who were beyond severe
Or
COVID.
is that the publication I saw? Maybe that's why it looks so good to me, because
if the control group was in there, you've seen the.
Okay. I thought that was the one they were objecting to. I'm like, it's all here. What are they objecting to? Okay,
yeah,
good.
No, the initial one didn't have a control group.
Okay, got it. Well,
Again,
it's in there. You got it. It's there. If they push back on that, they're irresponsible, frankly.
That's the one I forwarded to you, Dr.
Okay,
Drew.
got it. Okay, because I was reading, going, what are they pushing back on? It's all here. So I have a couple more questions. People are asking a lot of stuff on the chat here, so let me ask what they want to know. They want to know, is the Operation Warp Speed aware of leronlimab, or does that matter to this kind of an application?
They are. Cytokine received an email. I received an email from Operation Warp Speed. So yes, they are aware of it. And I think we'll move forward with them.
Okay. And the BCG, they're asking about the BCG on the thread. Totally unrelated topic. Do you guys have any opinion about the role that might have played or not played in the COVID?
Well, I don't know what role it played in COVID, but BCG is a very interesting immunologic phenomenon where you have, in bladder cancer, you can use BCG as a therapy for cancer, and it's nothing more than something that really irritates the immune system and soups it up locally in the bladder. And actually, we're doing a lot of work on that right now in another project in bladder cancer, which happens to have very high CCR5. And we have a project we're working on right now with a collaborator at Beaumont in Detroit, Shaheen Alani. And we're seeing CCR5 all over the place in bladder cancer. And then BCG we're using to treat bladder cancer. And it may be unique that that CCR5 is there, and that may be one of the reasons why it's able to attract the immune system, the immune cells that result from BCG therapy.
That makes sense, right? So now, so this is interesting. So RANTES now has a therapeutic role as well, right? You block it if it's overdone, and you can use it to attract activity.
Well, I think back to what Ram said in the beginning. Much like PD-1, PD-L1, you can either block PD-L1 with a drug, or you can be a PD-1 agonist. And so you have the ligand and the receptor.
Right.
And we've covered that in what we've done in InCell DX, covered both the receptor and the ligand, because you could conceive of two strategies for therapy: attack the ligand, neutralize the ligand, which would be RANTES. And I think, some of these newer therapies, like that drug from India and the new drug, the inhalation, the VIP drug, both decrease RANTES, which would have the net effect of blocking CCR5. And then, of course, there's a whole host of CCR5 inhibitors, including leronlimab, that are targeting the ultimate target in COVID, which is the CCL5 RANTES CCR5 pathway.
So let me just quickly circle back and explain what Dr. Patterson was talking about, that there are ways to block or enhance your immune system's ability to see things like cancer. There's where these CDL1, it's PD-L1 and PD-1 inhibitors or activators are sort of handshakes, the way the immune system sees things, let's say. Yeah. Is that a way to describe it?
I mean, those drugs, the cancer rate has gone down throughout the world, and immuno-oncology is a huge field right now. Those drugs that disrupt the PD-L1, PD-1 axis and restore our own immune response against cancer have been hugely successful. And that's what got me thinking about attacking COVID in the same way, by manipulating our own immune system. It was successful in cancer. Our body hasn't seen this virus, so we don't have a stored immune response against this virus. Let's use the most powerful tool that we have, much like we did in cancer with PD-1 and PD-L1 blockade, and attack it with our own immune system. And in fact, I think that's
what you're onto here. And let me state it again, if I didn't say it clearly enough, is that the cancer has to evade your immune system in order to grow. And we know what the receptors are in the immune system that allow your body, your immune system, to see the cancer again, so it can attack it on its own. I think that's the way to describe it. The VIP thing, is that a blocking of the binding of COVID to ACE2 or SARS to ACE2?
Yeah, so it's the vasointestinal peptide.
That's the Abitadel stuff, right?
Yeah, the Abitadel, right?
Okay.
Right. That's the one Dr. Patterson was.
Okay, got it.
So it's almost got two mechanisms of action. It's got kind of what I look at it as a direct antiviral, where it blocks that ACE2 receptor. That's the receptor where the virus is going to attack in the lungs. And that's something interesting, where it actually binds to that ACE2 receptor, so it blocks the virus from binding. And what's also interesting, it upregulates surfactant, because that cell, it's called a type 2 pneumocyte. It's a specialized cell in the lungs. It's only about 5% of the lung cells, but it makes this compound or this peptide called protein called surfactant. That's what you need for
It
gas.
allows the air cells to stay open. It's what if you put it on bubbles, the bubbles stay bubbles. That's sort of how it works.
Yeah, exactly. So it causes an upregulation of it, and it's naturally produced.
Well, they thought that was the mechanism of action. And the stuff I was reading, they thought, well, that's it. But I think that may be the minor mechanism, right?
So there, in the literature, there's immunomodulation, and when I started reading it, they put out I think yesterday they put out a preprint about 21 patients in Houston. Really interesting read. I'll post it up on Twitter for everyone to read it.
Right.
And they're talking about they've decreased interleukin-6. I think CRP, some of the inflammatory markers, but they haven't measured RANTES. And when I started reading, kind of diving into it, I found this article. I sent it to Dr. Patterson. I posted it on Twitter yesterday, where it's clearly stated that VIP inhibits the production of RANTES. So where leronlimab actually occupies that CCR5 receptor, we suspect maybe the VIP is blocking or inhibiting the production of RANTES. So it's almost like it's a different way, as Dr. Patterson said, of really targeting RANTES. So then I emailed Dr. Javid and I said, listen, I think you guys need to be testing RANTES. I got an email back 2 days ago and they said they're going to start looking into it now. So again, this could be proving Dr. Patterson's theory.
Yeah.
And the other drug Dr. Patterson mentioned was the drug in India, itolizumab, which blocks the CD6. That's a receptor between the macrophage and the T cell. And we found some literature suggesting that the anti-CD6 antibody, which is itolizumab, also blocks RANTES. It's in the literature. So I've emailed the researchers at Biocon and said, can you guys look at RANTES? I'm waiting to hear back from them. So we've got 3 drugs right now that all, like I said, all roads lead to RANTES. And who was the first person to say that? Dr. Patterson.
I get you. I'm getting lots of questions I want to ask Dr. Patterson. They're asking, can you explain the elegant way that leronlimab disrupts the CCL5 pathway?
Well, I mean, it simply is a competitive binder to CCR5.
Got it. Maybe rather elegant, we should call it just simple or direct.
Yeah, it's direct.
Yeah.
And in fact, we use fluorescently tagged leronlimab as a diagnostic tool to determine how much leronlimab is actually bound to the cells that it's supposed to be bound to. And that's been instrumental in dosing and determining, exactly what cell types you're inhibiting. Because like I said before, it's manipulating the cells that express CCR5 that's critical, and they all express different levels of RANTES. So you can conceive of using the levels of RANTES to help inform dosing for whatever indication that you're working on.
Interesting. I'm thinking about almost like we use pressors. We dial it up and down. It's kind of strange.
Exactly.
Susan, people are asking for the Twitter handles for our doctors. If you can stick those up there on the screen, we'd appreciate it. And then our buddy Andrew Ashkasvili, who I think first got me in touch with you, Dr. Yo. Test kits that recently got signed. Dr. Bruce.
Yes, we're very pleased to announce this morning agreement with M.D. Biosciences based out of Minnesota, a very skilled CRO and reference laboratory that specializes in flow cytometry, which is a technology that we use with our test kits that is ubiquitous around the world. And the great thing is they have labs in the EU where some of these new therapies are going. We know leronlimab just announced it's going over to the UK. And again, it'll give us access to, getting our kits into the reference labs in Europe through MD Biosciences, and give us a lab here to start testing as soon as is physically possible. And despite the fact that the drugs aren't out yet, we really feel that we can support these patients suffering these long haulers and help them with these diagnostics say, what exactly is going on? Because as we found in these long haulers, they still have elevated IL-6, they still have elevated IL-8, they still have elevated RANTES, they still have elevated TNF alpha, and it's a very heterogeneous group. Some have some of them, some have others, But we feel for you. There is something real going on. And I know, Dr. Drew, you know better than I, when people have chronic illness but don't necessarily know the source, it's almost worse. You'd rather just know what's going on. And of course, there's treatments, you know, for the long haulers that are available. But I think just knowing that they're not going crazy, that there is something real, there is something physiologic,
right?
Give people peace of mind that, okay, my body's still recovering. I can handle that, and may help with some of the mental aspects of recovery.
This phenomenon of long haulers has a history. The 1918 flu, there was a precisely identical phenomenon, and it was picked up by Dr. Menninger, the most famous psychiatrist of the 20th century, and he was struggling. Obviously, he didn't have these same technologies but he felt as there was some sort of ongoing inflammation of the brain, and he was seeing a wide spectrum of psychiatric pathology. Amongst them was something back in the day they called neurasthenia, which is fatigue. If you have a chronic, essentially a flu syndrome that keeps going, yeah, you're going to get depressed. And by the way, you know, immune cells and neurological material have a very close developmental heritage, and what affects the immune system tends to affect the central nervous system too.
Well, interestingly enough, we just had a patient's data come off the machine this morning, looking at immune cell exhaustion in a long hauler, 60 days out from Montana. You know, you would think, you know, breathe in the fresh air and recovery there would be fine. But you know what, the fact is, we looked at her immune cells, and they still had very high immune cell exhaustion markers. So her immune cell is exhausted. Still elevated interleukin-6 and interleukin-8, which gives you myalgias and joint pain, which is a common complaint. So, you know, we're making sense of what's going on in these long haulers, and then obviously that'll transition nicely into approaches to relieve their symptoms.
So two really good questions came up here. One was, are you using this? I guess there's some deployment for pancreatic cancer. Could you speak to that, number one? And number two, have you thought about looking at these inflammatory markers in chronic Lyme disease?
Well, that's a great point. I haven't, but I mean, there's another disease where there was a infectious insult, and following the infectious insult, you're left with months, if not years, of some sort of immunologic abnormalities that continue to just make you feel and suffer from it. And in fact, I mean, what COVID is also telling us by looking at this complex immunologic, you know, ballet, if you will, is that the immune system is real. The immune system actually can be very prolonged and can cause symptoms for a long time. So yes, Lyme is a perfect example. You know what? Pregnancy itself and postpartum depression, something that really got me thinking when I was looking at these panels, because there is this elegance to pregnancy where you're locally immunosuppressed in the placenta so you don't reject your baby, which is half foreign, right? So you're making all of these immunosuppressive cytokines like interleukin 10 and interleukin 4. All of a sudden you give birth and boom, your body shifts back to where it is, protecting the placenta, protecting the uterus, protecting the female genital tract from infection. And it goes back to more of a type 1 cytokine response with TNF alpha, interleukin 2, interleukin 12, interferon gamma. And there's no question in my mind that contributes to, you know, postpartum depression and these symptoms. And same thing with Lyme, and maybe same things with.
Makes perfect sense. Again, back to Dr. Menninger's observation in 1918, you know, and for the years following that too. And that was actually much like, you know, people are making a bit of the long haulers. It's not that common a thing, but that was one of the more serious consequences of the 1918 flu. Yeah. I have a philosophical question for you guys that's running through my mind. and this is a new topic. The three of us are biologists, each trained at very different stages in technologies. When I was trained, we were using restriction enzymes and acrylamide gels, and everything was inferential, everything. Dr. Patterson, you're the middle ground, and you're the group that really discovered this flow cytometry and all the things that you now use routinely, and not to mention the genetic, being able to code the genome. And then Dr. Ram, you're the newcomer. Do you have the perspective, Dr. Yogendra, on where biology has come from since I was being trained as a biologist?
Yeah, it's interesting. I always, Dr. Patterson and I, we've talked a little bit about it. I was probably probably in middle school, maybe even elementary school at the height of HIV. I mean, the Ryan White story. I remember being in elementary school, and that was really, really big.
Let me say, I was treating AIDS patients. Like that's all I was doing. That it was hand over fist. I worked in a county hospital, and that's all I did. I had either an oncology ward, infectious disease ward, even rheumatology wards were filled with AIDS patients. But go ahead.
I think what's interesting is with technology, it's easy now to get information. We can Google search, we can go on PubMed. I think from an information standpoint, it's much easier with the internet, but at the same time, there's also a lot of false narratives and people can come after you and try to discredit you very quickly, especially if you have, I think that's probably talking, when we try to discuss and try to figure out why are some of these, what Dr. Patterson is talking about, why is it only in a select group of people? Why aren't more physicians? It's shocking. The number of physicians that I've talked to have never heard of any of these things.
Do you know why? I think because we de-emphasized training scientists and then making them physicians. We said, no, we want poets and we want historians, which I thought at the time was a good thing. But you have to have, if you don't have that biological core, that way of thinking that the three of, even though we represent different generations of the evolution of biology in America, we think the same. And it's that thinking process that's so critical. That's sort of what I wanted to point out. Do you agree, Dr. Patterson?
I agree wholeheartedly. I mean, any lab can spit out numbers, but it's your ability to take those numbers and synthesize how they're impacting the patient and synthesize it into pathways and say, what pathway do I really have to attack to make this patient better? It is all about patient care, but what the fact is, I think you have to be careful to just not look at the outcome, but how did you get there? Because there's never going to be a drug that works on everybody. There's never going to be an approach that works on everybody. COVID is such a heterogeneous disease that there may be a number of different approaches. What was great about leronlimab was it had a multitude of effects on different aspects of the immune system. But the fact is that right for long haulers who may only have elevated IL-6? Maybe the arthritis drugs can come into play there, or maybe steroids can come into play there, or something that you can take more long term than some of these more sophisticated drugs that are needed to save lives. So I think you're absolutely right, Dr. Drew. I think we're in a area now where we have machines and AI, many spitting out numbers and analyzing numbers, but it's putting all of that together in a clinical picture and actually being trained in pathology. That's what we did. I mean, I think before my virology career, I was trained in classical pathology at Northwestern, and that's, you learn pathways, you learn the underlying reasons for symptoms, and you looked deeper into why somebody may be feeling a certain way.
But I would even argue there's a more fundamental thought process that I'm seeing in you, which is, and people are not trained to do this: hypothesis, experiment, data analysis, conclusions and weaknesses, and that's it. That's the scientific method hasn't been taught in a long time.
Yeah.
And that's automatic with you. I could see it operating.
Yeah. Well, it also gives credibility to outcomes. If you understand and you've planned going forward, if I do this, then that's my hypothesis. If I test by giving this drug and the outcome is aligned with the mechanism of action, that lends credibility, and you can resist some of the things that are out there for some other COVID drugs where the mechanism doesn't fit with the outcome, and there's always question marks, and it's really nice when they align.
Now you're talking about judgment, which is what comes late in the game after you've done this a long time, which is really what the skill is, right? But they're screaming at me on the chat about a paper on RLF 100. Either you want to comment about that?
Yeah, it came out yesterday. I think it was a 21 patient. It's not an EIND. They took extremely sick patients, the severe and critical patients. I think all of them were either intubated or on ECMO. ECMO is the life. It takes out oxygen. It oxygenates your blood outside the body, almost like a bypass machine. And they took these patients and they responded. I had Dr. Javid on my show last week. He's the CEO of NeuroRx, the company that is studying Abitadel. And it's very interesting because this drug is almost exactly like what Dr. Patterson is talking about with leronlimab. It has antiviral properties. Dr. Patterson's paper showed the decrease in the viral load. So with this drug, they're also showing a decrease in those inflammatory cytokines. And what's also interesting is they put these chest X-rays within 24 to 48 hours. There was dramatic improvement, and even a non-medical person could see that. When an X-ray, you see the white patchiness, and that's some sort of infiltrate, either an infection or some fluid in there. And then you see the X-ray from day 1 versus day 2, and you see a dramatic decrease. And it's pretty impressive. Just talking to Dr. Javid regarding that. So this is just an observational study. It looked at those 21 patients, very, very sick. They're doing a phase 2/3 trial right now at Houston Methodist. And right now, there's also a firefighter. I put his information up, his GoFundMe. Everyone knows I try to raise money for a lot of these COVID patients and cancer patients by selling Dr. Patterson T-shirts. So this firefighter actually, the family's just posting information. He was on 100% oxygen, and now after 1 dose of Abitadel, they brought down his FiO2 to 60%, which is pretty impressive if you're looking at less than 24 hours. That sort of response.
Well, it takes him out of harm's way a little bit.
Oh, yeah, absolutely. I do believe he's still on ECMO. He's still in the critical phase. But the study showed some interesting. There was some promise to that. They were able to successfully extubate some of the patients. I'm not sure what the number is right now. So that preprint just came out yesterday. But again, a lot of what's going on with Abitadel is the same thing that's going on with leronlimab and what Dr. Patterson has been talking about for the last 3 months. And Dr. Patterson brought up a very interesting point. A lot of people have been asking me, which is the better drug? Is it Abitadel? Is it leronlimab? If it's itolizumab, which one's the best? I go, we need more tools in our toolbox. And there's not one drug we can scale it to the level that we need to take care of not just only in the United States, but around the world.
Let me ask Dr. Patterson this, to that point, and now hearkening back to Dr. Patterson, your judgment. Do you have a sense, much like with HIV, we'll be using combination therapies? It seems like that's the way immune biology goes. Do you have any hunches under the hood there?
I appreciate that question very much, because what's interesting about the approach that we've taken is it's getting away from the virus, just like HIV. CCR5 inhibitors in HIV are unique in that they don't do anything to the virus, right? They protect uninfected cells from being infected and obviously preserving your immune system. What is great about that is they are not susceptible to the extreme mutation rate of HIV, so you don't have resistance. I think by the same token in COVID, we know COVID has significant sequence variation. And frankly, I think that's going to be the biggest challenge for vaccines and even neutralizing antibodies, is that, what we designed in the spring to be testing in the fall, by the time you get around to testing in patients in the fall, there could be significant sequence variation that is different than what you designed the vaccine or the neutralizing antibody against. So yes, I am very favorable on therapies that are directed at something other than the virus, because then you're going to be susceptible to what the virus does, as you suggested, in an evolutionary way, by having a high mutation rate and evading potential therapies.
So another way of saying that would be you're interested in your immune system being in charge.
Yes.
Yeah, bringing
Because
the immune system
I
back
think
online.
that's going to have less variability and less reliance on the viral sequence.
Yeah, it's interesting. I always think about, when I think about biological systems, I think about clouds. And when you're at a vaccine, it's a bow and arrow. You know what I mean? And our own innate biology is more set up to manage the cloud, the amorphous changes. Let me give you a chance to talk a little bit, Dr. Patterson, about cancer and where this might go in cancer therapeutics.
Well, again, I think the knowledge that we're much like HIV, which I think really spurred the explosion in molecular medicine and molecular diagnostics and all the tools that we're taking advantage of now in single cell biology. I think COVID has taught us a lot about the immune system and how we can manipulate the immune system. And I think because viruses and cancer use the same cellular-based immune response in a similar ways, I think we're learning a lot about cancer, how we can, some of the things that we looked at, like for instance, leronlimab seems to increase granzyme A, which are these little bullets that CD8s use to kill tumor cells or kill virally infected cells. It increases, granzyme A, which we put in the paper as well. So, and then the other thing is, if we're using CCR5 inhibitors for HIV and blocking infection of new cells, we've never asked the question, well, is CCR5 inhibition also enhancing the immune response against HIV-infected cells?
Right.
That are latently infected and replication competent, or even continue to replicate because they can. So it's got me to wonder, well, maybe we're not just blocking the receptor on uninfected cells. Maybe we're also enhancing the immune response against the already infected HIV cells. So I'm excited about the knowledge that has come out of COVID. This always seems to happen when you have these, these pandemics and tragedies that happen. You learn a lot about humans and biology and how we can exploit those to our advantage in other areas. And much like we learned from cancer about, hey, maybe using our own immune system in COVID is a good idea. And that's where I started, back in January in terms of my thinking.
And by the way, if you want to, you can see Dr. Bruce Patterson's TED Talk. If you want to lay this all out, it's all in the TED Talk. And I just go to TED Talks and you'll find his TED Talks there. Two things occur to me, and based on what you said, both are flying out of my mind as I speak. One is, you notice there a lot of people are on the chat going, well, does this work or does it not work? And then they're also expressing their criticism of Dr. Fauci. Do you notice how biologists and physicians talk? We don't say, this is all this or this is all that. We say the evidence suggests right now, we're enthusiastic about, it's looking good. We're not saying it must be used, it has to be. We're saying this is science. This is an evolving process. We're moving towards the truth. We're moving towards more advanced therapeutics. And this, as Dr. Patterson said, this tragedy accelerated all that. So you don't go, this drug works, we must use it, it's not how we operate. It's not how science goes. It's not how clinical expressions go. The other thing is I'm on the board of the Prostate Cancer Foundation, which PCF.org, anybody's interested, it's the leading funder of research on prostate cancer and several other cancers. In fact, we have some of the stuff that is really, all of the immune therapies you were talking about, of course, we're involved with, and we are looking at stuff for colon cancer too. But just this morning in a long call, we were talking about how COVID had changed the landscape for us too.
Yeah, and by the way, urothelial tumors, bladder, prostate, have the highest levels of CCR5. And we're writing a paper again with our colleagues from Beaumont in Detroit on CCR5 expression in urothelial cancer. So that may be another relatively safe approach, along with the PD-L1 inhibitors to attack bladder cancer and even prostate cancer. So
and again, this is a perfect, what I was talking about earlier. We hit it from two. We hit the PD-L1 inhibitor and the CCR5. We were hitting it the 1-2 punch, which something about biological systems. That's when you really get massive change, is when you can hit it at two sites, because it kind of finds its way around you if you're just using the bow and arrows we were talking about earlier.
It does. Yeah.
Well, gentlemen, it has been a profound honor. Dr. Yo, thank you for bringing Dr. Patterson in here. I cannot thank you enough. And I enjoyed, Ram, our conversation last time as well, where you really laid out the science. I hope today people had a chance to hear more about this. And, of course, Dr. Patterson's amazing work. Go see his his TED Talk. It's probably available on YouTube for that matter. But I'll give each of you sort of a wrap-up. Chance for wrap up. Dr. Yogendra, you first.
Yeah, well, first of all, thank you, Dr. Drew. And again, I always thank Dr. Patterson. And every time he speaks, it's always a medical science lesson, even for me too. So thank you, Dr. Patterson. I think it's important for everyone to remember that, it's great talking about the science and medicine. I mean, it seems like we're all three of us are science junkies, but it's about making actionable plans. And it's about letting the public know that there's a lot of doom and gloom and scary, people, there's so much of anxiety. We talked a little bit about the psychological aspects of the pandemic, but there are physicians and the researchers like Dr. Patterson and many others who are really, really just doing a lot of work. The question is, what do we do next? This is great. We can talk about this. We have your viewers, our viewers, our little circle, but we got to figure out an actionable plan so that let the American public and let really the world know that these people like Dr. Patterson are out there doing these type of research. They're really thinking outside the box. And it's just about, I'm not sure what next and how we can get this word out, because look, it's not quack. It's not crazy science. This is grounded in the basics of science and medicine. So really, what do we do as a group? I don't know, maybe you can help out, Dr. Drew. Obviously I'm trying
to get the word out. That's all I can do is push. I feel like I'm the French underground in the middle of a war, pushing stuff out, because there's so much craziness in the general press. So whatever I can do to support you guys, please let me know.
Yeah, no, and again, we really appreciate, because sometimes you feel like you're on an island all along, alone. And when I got introduced to Dr. Patterson, I've told this many times to him, wow, I feel you're the only doctor I've talked to, one of the only few that doesn't make me feel like a crazy person. I can run my crazy theories, text him and email him hey, Bruce, what do you think about this? And he's very supportive. And so we need the American public to know about Dr. Patterson and his research. And by the way, I don't have any financial business relationship with IncelDX or Dr. Patterson. I just have absolute respect for what he's done. So my final thought is, let's try to figure out a way to get this research out there. Let the public decide. If they want to decide, hey, listen, this is junk science, then let the people know. But they need to know what's going on so that they know there's some hope that really smart people like Dr. Patterson are out there trying to save lives.
And Dr. Patterson, final words. We have on my chat, I've got people that want to write a screenplay and cast Matt Damon as you in the movie. So tell us, give us your last thoughts.
Can I be in the Bourne movies then?
Exactly. This will be the viral Bourne.
That's right, viral Bourne. But first of all, I'd like to thank Dr. Yo. I mean, he's brought us all together to have respectful dialogue where we all listen to each other's points of views. As you said, Dr. Drew, there's no one point of view. I think we have to join together in this pandemic and share our thoughts, share information in a respectful way, and remove the politics, and even step outside of our companies at times and just say, hey, we're trying to save lives, we're trying to save people. I have great satisfaction knowing that we probably already have saved a number of lives, and we have to continue in that vein. And Dr. Drew, thank you for your support. I mean, I've always been a fan because I've always thought, every medical condition, everything that we go through, there's a mental aspect that at times is either discounted, disregarded, or not understood. And I think we have to think of the body as a whole, and not the body as just a bunch of different systems and compartments. And I think what you've done and what you are doing is amazing in coming together with a more holistic approach to human health. And I think
I appreciate that. It's really because I'm an internist by training, but I worked in a psychiatric hospital for many years, and so the whole picture evolved for me. In the morning I was walking into an ICU, and by noon I was walking onto a drug unit or into a depressive disorders unit. So I got to see the whole human experience in a way that people aren't being trained in that anymore, unfortunately, and I'm worried about that. That's why I'm trying to download everything I've seen. But it all started, though, I have to say, with a really good foundation in biology. Like that's the thing I noticed about the three of us. I could see it. I know it. I know how your brains operate. And we just don't do that anymore. And I think that's where a lot of the craziness is coming in the media, that you have people that just learn how to pronounce a medication or a molecule, and they have an opinion about it. That's totally insane. They should shut their pie hole. Or go study some science for real, or get a background in or something. I don't mind people having an opinion, but having these vehement opinions based on I don't know what. But again, I digress. Gentlemen, it has been a true pleasure. I thank you for being here. If I can do more, if there's stuff you want to push out, if there's other people we should be talking to, please let me know. And I hope to stay in touch with you guys. And I thank you so much.
Please, thank you so much.
All right, you guys.
Thank you, Dr. Patterson. Thank you.
And Susan, maybe we can tweet out the link to Dr. Patterson's YouTube, TED Talk. People are asking for that. The rest of you, thank you for being on the chat. Thank you for being here this afternoon. We really, really appreciate it. That's an interesting idea. Someone wants a Netflix series about real bioscience. Hopefully that could keep people's attention. That's a great idea. People are thanking my guests. I see you on the thread here, and I appreciate you all being here. Jiro wants me to shut my pie hole. I'm not sure I'm going to. There was a lot of action today about Dr. Fauci on there. Think about how you heard the three of us talk, and then sort of listen to how that's how he talks. He talks in sort of based on the best available knowledge right now. Here's what I think, and then he'll shift. Maybe we'll find out that Adaptogel or lorlatinib doesn't work or has some terrible side effect. Then I will adjust my opinion. Right now, this is interesting science that's getting us to open up a piece of the immune system that I wasn't even trained in. They didn't even know existed when I was in medical school. These are massive changes in medicine. Now, my personal opinion is you hear Dr. Patterson has a vision for changing cancer and bladder cancers, and indeed he'll get there. But right now, I'm telling you how we approach the treatment of the critically ill patient changes immediately. It's already changed and going to change forever because of what we've been through here. Susan, maybe we ought to get a vaccine kind of guest in here. People seem really interested in these biological guests. The vaccine story is kind of an interesting thing as well. But again, you guys send us your suggestions. Send it at contact@drdrew.com. We appreciate our friends at Hydrolite for sponsoring us today. It lets us keep doing these things and have all this fancy technology. It lets me continue the French underground and continue pushing things out. We have, again, I don't have any interest. If I do have an interest in any of the things we talk about here, I will tell you. I mean, obviously, Hydrolite sponsors this show. but I don't know anything about the companies that are involved with the stuff we were talking about today. So be that as it may, I thank you so much for being here. And we'll, Susan, we are in here tomorrow at what time? I can check the schedule. I should check rather than hoodwinking you like that. I see it as 12 o'clock tomorrow. And we are going to have Dr. Deborah So on soon. I don't know if you heard her. Is that tomorrow?
Tomorrow's Friday, honey.
Yes. I see noon.
We don't have anything tomorrow.
Oh, it's on the schedule for
Did
noon.
you change your hair appointment?
I did.
Okay, maybe noon.
So noon, maybe we'll just take some questions and stuff ourselves. But I think my next guest can be Dr. Deborah So, and she is she's the gender academic who got in a lot of trouble on the Rogan show, and
Actually, we have your daughter in here on Sunday.
Oh, and on Sunday, my daughter's going to come in. She's doing a project on cults. We're going to talk about that. And again,
we also have Dr. Donovitz coming to talk about bioidenticals.
Great. So Donovitz is on Tuesday.
And then hopefully Dr. So after that.
Yeah, people have lots of questions about the bioidentical hormones, and
we got a lot of good doctors.
We got a lot of good stuff coming up. And if this is what you like, let us know. Again, contact at drdrew.com. Let me look quickly through your questions here. Is there anything? You do enjoy the doctors as a guest. We're going to wrap this thing up. Thank you all for being here. We'll see you tomorrow.